control of the dorsoventral axis and the anteroposterior axis, among other things, in the flies. And she had come across a certain number of genes which, when mutated, would lead to aberrant embryonic development. And so she gave a certain number of names to those genes, and the genes later, when they were cloned, turned out to be part of the cascade, which is shortly illustrated here. One of the central genes which she identified gave a phenotype of embryonic development which she found extremely bizarre. And in German, you would refer to that as Toll, and she called the gene product, she called the gene Toll, and later on, when it was cloned, it turned out that it was a coded for a transmembrane protein. Now, I'm not going to detail that because my time is too short, and so I'm just going to summarize in the coming slide what we found over the years. And these were many years of work in the laboratory by very, very active scientists of great talent, namely in molecular biology. That was the role of Jean-Marc Reichert. In molecular genetics, it was Bruno Lemaitre, and some of their colleagues. And then, as we'll see, other persons. I'm going to show the slides, the figures, the photographs in the end. And so what I'm summarizing here is that it appeared in the beginning that we had fungi and gram-positive bacteria interacting somehow via Toll and then activating NF-kappa leading to the production of antifungal and some antibacterial peptides. Now, it later appeared that this pathway was not responsible for the production of the antibacterial peptides of the type of deutericin, which was the one we had first cloned in our hand. It was the first gene we had in our hands in the system. But another distinct pathway was responsible for this. So gram-negative bacteria activate this pathway, which we refer to as IMD for immune deficiency. Also, again, in the cell, activating NF-kappa via distinct family members. So in the fly, they are three NF-kappa family members. And as you see on the left side, we have dorsal and leaf controlling antifungal responses. On the right side, we have relish controlling the typically antibacterial, anti-gram-negative bacterial responses. So this then led us to probe if these pathways played a significant role in the survival of the flies, to really show that we were dealing not with simple laboratory experiments, but with something which was relevant for the life of the fly. And here we show Toll pathway mutants, which are sensitive to fungal infection, experimental fungal infection. And as you see, the wild type, as I say, the normal flies, survive relatively poorly, but survive, whereas Toll mutants, half of them have already disappeared after 2 days. Conversely, IMD pathway mutants, remember, which affect the transcription of the antibacterial peptide genes, those mutants, when infected with E. coli, die away very quickly in opposition to the wild type, that's to say, the normal flies. And this was published in our paper with Lemaitre, Michaud, Reichert, and Nicolas. And also in that paper we showed a fly which had been infected by Aspergillus, and we concluded from our data, which was still relatively fragmentary but clear, we concluded that the dorsal-ventral regulatory gene cassette, the one which had been identified in early embryos as maternally expressed genes, that this controls the potent antifungal response in Drosophila adults. Now, let me go a little bit further and say that, summarize microbial inducers and the cognate receptors in the innate immune response of Drosophila. That was the other aspect, the other question which we asked, to which we come back. Now, again here, because of lack of time, I'm not going to show the evidence. This has been, of course, published. And I'm just showing here the conclusion. And it is important for us, in a phylogenetic perspective, to keep in mind what I've been saying in these 2 points. Number one, bacterial peptidoglycans, which are present in all bacteria, are recognized by dedicated circulating or transmembrane proteins, referred to as peptidoglycan recognition proteins, which have evolutionarily derived from amidases. And the second point is that fungal beta-glucans, which are present in all fungi, are recognized by circulating proteins referred to as glucan-binding proteins, GNBPs. I should mention that there are 13 PGRPs in the fly and 3 GNBPs again in the fly. And I should also mention that our contribution here was through mutagenesis screens to show, to identify genes, and many structural studies have since been done by other colleagues and in which we had not been involved. Now, I'm summarizing this information as we had it after, again, quite a large number of years. We understood that fungi react with dedicated circulating binding proteins, referred to as glucan-binding proteins. They activate a proteolytic cascade, an amplification cascade, lead to the cleavage of a polypeptide, circulating polypeptide, which is referred to as Spätzle, again in reference to the phenotype which had been identified by Mr. Fuller. Now we know now that Spätzle, that is a totally different opening now. Spätzle is a neurotrophic, is involved in development. Now, activation of toll and then effector genes. On the other hand, gram-positive bacteria interact with a dedicated protein in the hemolymph, which is the gram-positive peptoglycan-binding protein, also activating proteolytic cascade, which has been deciphered since, and activate the toll pathway. On the other hand, the gram-negative bacteria interact with other dedicated peptoglycan-binding protein, which is distinct from the one of the gram-positive pathway, and activate then the IMB pathway via a transmembrane receptor without any intervention on amplification cascade. Okay, let us now go to part 3, from insect to mammals. I'm speaking here about the years 1992 and following. The context was that at that time innate immunity was believed by some scientists to be required to boost adaptive immunity. And among those, one of the proponents was Charlie Janeway, who was professor at Yale University, whom I had contacted in 1992, and Alan Zykowicz, I'm going to show the pictures in a moment, Alan Zykowicz, who was at Harvard Medical School at the time. And so they believed that innate immunity was absolutely required to get a potent adaptive immunity. But this was not a general belief in the community yet. So let me show the context. Again, here more clearly, this is Ralph Steinman, who shared the prize in 2011 with Bruce Boyd and me. And so we're showing here a dendritic cell, which are cells which are really the central sensors of infection in mammals. And they react via hypothetical receptors, it was believed, it was not identified, to microbial fragments, and in response activate NF-kappa B, and NF-kappa B then leads to innate immune responses, for instance antimicrobial peptides, which had not been clearly demonstrated in the mammalian system at that time. It was believed that production of antimicrobial peptides in the mammalian system were constitutive, which turned out to be too restricted, and activation of the adaptive immune response, it was believed, and this would go via, for the people in the audience familiar with this, it's classical antigen presentation, co-stimulation, I'm not going to this, and production of many specific cytokines which would activate the adaptive immune system. So in other words, we are here in the presence of lymphocytes, which will give a strong adaptive immune response, but to do this, they need to be stimulated, was the hypothesis, by the innate immunity, and for which there was a rationale in the story. You know that for at least those in the audience, I guess most of the persons here know that the lymphocyte repertoire of those lymphocyte cells is so enormous, and they can recognize any peptide or polypeptide which can occur in the blood. So if there's a microbe appearing, the naive lymphocyte would not be able to distinguish between a peptide from a microbe or a self-peptide. So there was somehow someone had to tell, that was the way Charlie Janeway in the discussions would express himself, someone had to tell the adaptive immunity, that the peptide which is appearing now in the dendritic cell via this hypothetical receptor, that peptide signals an aggression. For that reason, they said, the number of those receptors should be relatively low, not in the millions, but maybe dozens. And they should recognize modules, ligands, as we would say, which are typical for bacteria or fungi, actually do not change your evolution. Now, you see